Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. So we talked about the alternative pathway in the previous video lecture. Now this is another video lecture which is also regarded to the complement system. And today in this video lecture we are going to talk about the last pathway of the complement system, which we called as MBL pathway. Okay. Now in the previous video lecture, if you guys remember, we talked the alternative pathway activation. So before going to explain the MBL pathway, you guys need to understand about the alternative pathway and the classical pathway, because both alternative and classical pathway are actually similar to the MBL pathway. Okay, so once you guys get idea about the alternative and classical pathway of the complement system, then the MBL pathway will be very easy for you. Okay, so let me erase this one portion and let me start. So let me write, or we can say MBP. Now this MBL stand for Mannose binding lectine, and this MBP stand for Mannose binding protein. So we can also call that MBL, or we can say that MBP. Okay, now in this pathway, what actually happen? We have the Mannose sugar molecule, which are usually bind on the bacterial surface. Okay, Mannose are actually the sugar molecule, or it is actually the carbohydrates that are usually present on the bacterial cell membrane. And what are actually the lectine? Lectine are the proteins that usually bind to the Mannose sugar molecule of the bacterial cells. Okay, now what actually happened in the previous video lecture, if you guys remember, we talked that in alternative pathway there are the antibodies involved, and in the classical pathway there are still the antibody are involved, but in this case we doesn't need any antibody. Okay, now let's suppose the bacterial cell are get internalized. This is always activated against the bacteria and also activated against the viruses, because on the viruses envelope there is also the Mannose sugar molecules or Mannose carbohydrates are also present. So we can say that against the viruses, against the envelope viruses, this one pathway is also activated. Okay, now let's say in this case we take the bacteria. So let's suppose the bacteria are get internalized. Let me draw. Okay, on the surface of the bacterial cell they have the antigen, but in this case let's suppose they have the Mannose sugar molecule, or which are the specific carbohydrates. So let's suppose these are the Mannose molecules or carbohydrates. Okay, now this carbohydrates, due to this way a protein is activated, we usually call it lectine protein. But before that, there is a question arise in our mind that how these proteins are activated, which kind of the cells are usually responsible for the activation of these lectine proteins. Okay, now for the lectine protein activation, what actually we have a cells which we usually call that macrophages. Now macrophages is usually a part, all of you better know that we talk that it is a part of innate immune system. So this macrophages is activated. Let's suppose, let me draw the cells here. Let's say this is the macrophages. It is activated against bacteria, viruses. Let's say in this case it is activated against the viruses. Now after that, this macrophages engulf these viruses. Now after some time they digest the virus, and after that this macrophages secrete chemicals. Now they will secrete the chemicals. Now let's say these dots are representing the chemicals. These chemicals will stimulate the liver cells. So they will stimulate liver cells. Liver cell stimulation. Now once the liver cells are stimulated by each of the chemical which are secreted by the macrophages, after that these liver cells then produce the lectine proteins. So let's say this is the lectine proteins. Lectine. Now once we get the lectine proteins, these lectine proteins are bind to this Mannose sugar molecule which is present on the bacterial surface. So let's say this is the, now this is the lectine. Now once lectine proteins are bind, this lectine proteins are usually against this Mannose. That's why we can call that membrane binding. We also call it membrane binding protein because, no sorry, we also called it Mannose binding protein. This lectine is also called as what? Mannose binding protein. Try to remove the spelling mistake by yourself. Now what actually happened? Once this complex are formed, then we have an enzyme which is activated in this case, and this enzyme is called as Masp. So let me write. Now what does this Masp stand for? M stand for MBL, A stand for associated, S stand for serum, P stand for protease. Now this enzyme can cleave the C4 protein. If you guys remember in the previous video lecture, we talked the C4 protein, we talked the C2 proteins. All these are actually the complement proteins. Now once the C4 proteins are activated, let's say we have the C4 proteins that are activated. They have again the two subunit. If you guys remember, the one is the larger and the other is the smaller. Now what actually happened? This is what, this is the C4 proteins. Now this C4 protein touch this one enzyme. Let's say they touch this enzyme. Now once they touch this enzyme, or once they bound with this enzyme, after that this C4 are break down or divided into two fragments. One is the large fragment, other is the small fragment. And if you guys remember, we talked that the large fragment of the complement protein are acting as a help in opsonization process, and the small fragment of any complement system help in the they are help in anaphylatoxin. Okay, so what actually happened, this C4 protein are converted into two parts. One is the larger and another the smaller. So we get the larger one. So let's say this is the larger one. Sorry, let's say this is the smaller one, which we call it C4A, and the C4B are bind with the cell membrane with this complex, mean lectine Mannose complex. With this lectine Mannose complex, the C4B proteins are bind. So let me draw. So let's say this is the C4B protein. Now after that, once it is bind, so then after that another protein is activated which we call that C2A proteins. Okay, sorry, C2 proteins, C2 proteins or complement 2 proteins. Now again this complement 2 proteins will touch or associate with this enzyme for the cleavage. This enzyme is act as a cleavage process. They hydrolyze the complement proteins. Now again we have the C2A proteins are active, C2 proteins activated. So let's say this is the C2 proteins. Again this C2 proteins touch this one enzyme. Now after some times this C2 are then converted into two fragments. One is the larger, other is the smaller. Now in this case, if you remember, we talked in the in the classical pathway, if you remember, we talked that the smaller subunit are the B and the larger subunit in this case are A. Okay, so this is the smaller one, which is the C2B, and the larger fragments of this one protein attach to this cell membrane of the bacteria. So this is actually C2A. Okay, after that this complex act as a C3 convertase. In the classical pathway, if you guys remember, we talked that C4B, C2A acting as a C3 convertase. Similarly in this pathway it is also acting as a C3 convertase. Okay, and so that's why I am advising you guys that you guys must watch the classical pathway, because the MBL pathway is similar to the classical pathway. Now once this complex are formed, we get the C3 convertase. Now in this case the C3 proteins are get activated, and this C3 protein are then cleaved by this complex. So we get the C3 proteins. So it is cleaved by this one complex, which we call a bimolecular complex or C3 convertase. Now in this case, the larger subunit is bound to the cell membrane with this complex, and the smaller is released out. So this is the C3b, and the C3a is released out, which act as an anaphylatoxin. Now after that we get trimolecular complex. Now this trimolecular complex is acting as a C5 convertase. Sometime what actually happen in C3 convertase, C3b protein covered the whole bacteria, and after some time, in the previous video lecture, if you remember, in alternative and in classical pathway, we talked that once the bacteria is covered with the C3b proteins, so they will activate the this C3a activate the mast cell. Mast cell are degranulated, and they will secrete the histamine. Histamine will dilate the vessels, and after some times they will activate and attract the neutrophils and macrophages. Neutrophil and macrophage finally engulf that. But sometime what actually happen, this C3 protein will just attach, and let's say they will not activate the mast cell, or not other cells are activated. Then after that what actually happen, we have the C5 convertase. Let's say this trimolecular complex act as a C5 convertase. So after that we get the C5 proteins. So C5 proteins again, let's say this is the C5 proteins. C5 proteins will comes and cleaves by this one. This trimolecular complex is this time acting as an enzyme. So they cleave this C5 proteins. This is the C5 protein, and this is the C3. C5 proteins are cleaved into two fragments. One is the larger, other the smaller. Now the smaller is released out, which is C5a, and the larger are bind. So let's say this is the C5b. Okay, now okay, one thing, this C5b are not bind. Okay, sorry, C5b are not bind. Remember, in the previous video lecture, if you guys remember, we talked that in the alternative and in the classical pathway, we talked that C5b are not bind. They are just circulating around this bacterial surface. Now how they will bind to this, or how they will embed to the cell membrane? They will be embedded or attached or covalently bind to the cell membrane of the bacteria when they associate with some other complement protein, which we call that C6, C7. So let's say we have another proteins. Let's say this is the C6. Then we have C7. Now what happened? C6, C7 are activated, and they will just covalently attach this C5b. Now once C5b are attached, then after that C8 protein is activated. So let's say this is the C8 protein. C8. And if you guys remember, we talked that C8 protein is acting as a transmembrane they are usually responsible for the transmembrane activity. So they will embed here these two proteins C6, C7 associated with C5. They will just bind this C5 covalently to the cell membrane, but the C8 will embed the whole complex. The C8 will embed the C5b, 6, 7. The whole complex they embed. Okay. Now after some times, a very small pores is created inside the bacterial cell membrane, and after some times we get another protein which we call that C9. So here the C9 proteins are activated, and they make a hole. So let's say this is the C9 proteins. Now after some time the C9, this is the C9 proteins. Now once the a very small hole is created due to this C8 proteins. The holes is further more larger and more bigger and more facilitated due to this C9 proteins. So remember, in the alternative and in the classical pathway, we talk that the same protein are not present only at that area of the bacterial cell. The same protein are also present on almost all part of the bacterial cell. So we get a lot of pores on the bacterial cell. Okay, and due to this way, the ions and the water molecules are get internalized. Let's say these are the ions. These green dots represents the ions, and these black dots represents the water molecules. They are get internalized. Due to this way, the bacteria are swell up and die. Sometime what actually happen, this bacteria will release out all the cytoplasm and all the important ingredients, due to which the bacterial cell are shrink and die. Okay, so this is how this MBL pathway will kill this one bacteria. Okay, now the remaining small the remaining small portion of complement proteins, in the previous video lecture we also talked that each proteins are acting as a anaphylatoxin. Now they will activate, among them this C5a will activate macrophages. If you guys remember, we talked that they will activate the macrophages and the neutrophils. Macrophages and neutrophils will engulf this bacteria. And this C3a, we already talked that it activates the mast cell. Okay, now again in the previous video lecture, if you guys remember, we talked that in the alternative and in the classical pathway we need antibody. Okay, in the classical pathway, if you remember, we need IgG antibody, and in the alternative pathway we need IgA and IgE antibody. But in this case we don't need any antibody. Autoantibody are also protein, but in this case we need a protein, we call that lectine. Okay, so that's in a sense about the MBL or MB pathway. Okay, we also call that Mannose binding lectine or Mannose binding protein, Mannose binding lectine pathway. Okay, I hope you understand about this video lecture. If you have any kind of questions related with the complement system, which contain the fourth video, so write the comment, write your question through comment. Inshallah, I will give you answer as soon as possible. So like all the videos of the complement systems, and share all these complement videos lecture to your friends, and must subscribe the channel, because in some other video lecture we are going to talk about some other topics which are related to the immunology in detail. Thank you so much for watching this one video.